At the end of our last video, we started to talk about gas transport, specifically oxygen from the alveoli into the blood. Because of the nature of alveolar and blood vessel or capillary construction, the thin walls of these structures make it easier for oxygen or carbon dioxide to pass from one structure to another. Now, as it turns out, oxygen is not very soluble in blood plasma. So, in order for it to be transported through the blood to and from tissues, oxygen requires a little help in the form of hemoglobin. And we all know that hemoglobin is an important molecule that's found in red blood cells. And hemoglobin is an important molecule that's found in red blood cells. Now, we learned about hemoglobin previously. Hemoglobin has 4 protein subunits. Each protein has a heme molecule as well as an iron atom. And it's the iron atom that helps to transport oxygen. Well, in order to understand gas transport through blood, it's also important to understand gas behavior. So what we're going to focus on is the behavior or the relationship between oxygen and hemoglobin. So we know from our previous discussions that the 4 subunits of hemoglobin can accommodate 0, 1, 2, 3, or 4 molecules of oxygen. So depending on the oxygen saturation of blood, that will determine how many oxygens are bound to hemoglobin. So we're going to take a look at a relationship between the partial pressure of oxygen and the percent saturation of oxygen on hemoglobin. So the graph that you see here is described as the oxygen hemoglobin dissociation curve. So let me explain a couple of things here that this graph illustrates in terms of the relationship between the partial pressure of oxygen and the percent saturation of hemoglobin. So on the x-axis of this grid, of this graph, we have the partial pressure of oxygen. If you recall, partial pressures are a function of the total pressure, and we described that as Dalton's law previously. So the partial pressure of oxygen, depending on sort of which part of the circulatory system it's located in, has different partial pressures. On the y-axis of this graph, it shows the percent saturation of hemoglobin. So this relationship between partial pressure of oxygen and saturation of hemoglobin has a slight curve to it. So it goes up very steeply at first and then starts to level off. So if you recall, the partial pressure of arterial blood equals roughly about 100 millimeters of mercury. So if we were to measure the saturation of hemoglobin at this partial pressure, we would find that nearly 100% of the hemoglobins in arterial blood have oxygen bound to them. So this is in arterial blood. All right, well, as we know, tissues need oxygen in order to generate ATP. So once the oxygens have been unloaded from the hemoglobin to the tissues, there will be less oxygen on hemoglobin. So if we were to measure the percent saturation of hemoglobin in venous blood, we would find that the partial pressure of oxygen in venous blood is approximately 40 millimeters of mercury. So if we use our graph there, what we find is that at 40 millimeters of mercury, the percent saturation of hemoglobin is approximately 75%, which is still quite saturated. So the main difference between arterial and venous blood in terms of saturation only decreases about 25%, about 1/4. So when you think about it, there is still quite a bit of saturated hemoglobin even in venous blood. All right, so that might not sound too significant. However, what is dramatic is that this occurs at resting levels of activity. But if you decide to get very active, go out and run or participate in some exercise, or where metabolic demands are significantly higher, particularly for oxygen to make ATP, the hemoglobin has the ability to unload a good portion of the remaining oxygen. And if we look at this graph here, what we saw previously is that the reduction in oxygen saturation of hemoglobin declines about 25%, about 1/4. However, if we look at the offloading of oxygen under stressful conditions, what we find is that there is a dramatic decline in the percent saturation of hemoglobin under stressful metabolic demands. So hemoglobin has the ability to offload a significant amount of oxygen, should that be needed, and from 75% saturation, that can go as low as roughly about 30% saturation if needed. So hemoglobin can offload a lot of oxygen very quickly if it has to, if metabolic demands require extra oxygen in order to maintain metabolism. So the oxygen dissociation curve helps to show a relationship between the partial pressure of oxygen and the percent saturation of hemoglobin. So one of the things I would expect you to do on an exam is, if you were given the partial pressure of oxygen, I would expect you to be able to determine the approximate saturation of hemoglobin. So let's do one quick example here. If the partial pressure of oxygen is 60 millimeters of mercury, what would be the percent saturation of hemoglobin? So in order to determine this, the first thing we need to do is draw a vertical line at partial pressure of 60. So let's draw a roughly vertical line until it touches our dissociation curve. Then what we need to do is draw a horizontal line over to the y-axis. So at a partial pressure of 60 millimeters of mercury, the percent saturation would be approximately 90%. So 90% of the hemoglobins would be saturated with oxygen. So that's quite a bit. So one of the more important roles of hemoglobin is to transport oxygen. It also transports carbon dioxide, but we'll talk about that here in just a little bit. However, the binding of oxygen to hemoglobin can be influenced by a number of factors. One factor that influences how well oxygen binds to hemoglobin is body temperature. The higher your body temperature, the more oxygen will be discharged from the hemoglobin molecule. Another factor that determines or has an influence on the binding of oxygen to hemoglobin is the partial pressure of carbon dioxide. So the more carbon dioxide that is present in the blood, the more likely oxygen will be discharged from hemoglobin. Also, too, a factor that influences how efficiently oxygen binds to hemoglobin is the amount of acids that are present in blood. And we simply describe that as the pH. So the lower the pH, or the more acids that are present, the more likely oxygen is going to be discharged from hemoglobin. All right, so let's take a look at this in terms of the oxygen dissociation curve that we just looked at or considered a moment ago. So up at the top of this diagram, we have a red blood cell with some hemoglobin and some oxygen attached to that hemoglobin. As the hemoglobin discharges oxygen to the tissue cells, we have our dissociation curve as we saw it previously there. So under normal body temperature, normal levels of PCO2, and normal pH levels, our oxygen dissociation curve remains the same. So no changes to that curve will take place. However, when we have higher levels of carbon dioxide or higher levels of hydrogen or acids present, or if the body temperature increases, that causes a change in the relationship between the partial pressure of oxygen and hemoglobin saturation. So let's take a look at the change in that graph that occurs with these changing conditions. So over here in the lower right of this diagram, we started with the normal oxygen hemoglobin dissociation curve. But an interesting thing happened. What happened is, with the extra carbon dioxide, extra acids, and higher body temperature, we find that oxygen does not bind to hemoglobin as readily. And in fact, hemoglobin will discharge more of its oxygen under these certain conditions here. And that changes the curve. And so what happens with the curve is that the curve shifts down and to the right, down and to the right. So we started out here with the dashed line here, which was our original curve. And so because oxygen cannot or does not stay bound to hemoglobin as tightly as it did, the hemoglobin will discharge the oxygen. And so that shifts the graph down and to the right. So this type of behavior with the higher oxygen unloading is referred to as the Bohr effect. The Bohr effect. So when body temperature is higher, when the levels of CO2 are higher, like say with exercise, and if production of acids should increase, all of those conditions will cause hemoglobin to discharge oxygen and not bind as tightly to it. So let me try to describe this for you with this particular description. It's not a perfect model, but I think it will be helpful. Pretend for a moment that this school bus is hemoglobin. And on this particular school bus, we have 20 seats for 20 students. And this bus can only carry 20 students, but we have a number of different students that need to ride this school bus. And as it turns out, there are 40 students that need to ride the school bus, but only 20 can fit on the bus. So, if, for example, one morning, 15 of the oxygen students showed up at the bus stop, and 5 carbon dioxide students also showed up at the bus stop, those 20 students could get on the bus and ride comfortably. So, because there were more oxygen students on the bus, that prevented any other carbon dioxide students from riding on the bus. So, the bus makes its way to school, drops off the students, and so the bus brings the students back home from school. Now, if on the way home, if on the way home, 15 So somehow the other students had to make their way to school in some other fashion. So if at the end of the school day, the students needed to ride the school bus home, and say 15 carbon dioxide students got to the bus stop first, and say only 5 oxygen students arrived before the bus had to leave, then no more oxygens could get on board the bus because, well, more carbon dioxide students were able to get on the bus first. So to kind of summarize this model here, when there is more oxygen available, the hemoglobin can accommodate the oxygen, but that prevents the carbon dioxides from being transported on the hemoglobin. Conversely, if more carbon dioxides are attached to hemoglobin, that prevents oxygen from binding to hemoglobin. So both oxygen and carbon dioxide are competing for spaces on the hemoglobin bus. And so the Bohr effect crowds out oxygen molecules from attaching to the hemoglobin. And so that causes the graft to shift down and to the right. So looking at our graph one more time here in the lower right part of the screen. In principle, the graph can also shift up and to the left as well. If body temperatures are lower and carbon dioxide is also low and acids are low, that also causes a shift in the graph up and to the left. So under those circumstances, we would have a graph that looks roughly like that. And in fact, this type of shift in the graph is called the Haldane effect. So, as I mentioned previously, oxygen and carbon dioxide are both competing for places on the hemoglobin. So, depending on which one is more abundant, will prevent the other from binding to hemoglobin. All right, so let's talk a little bit here about carbon dioxide. Carbon dioxide is a metabolic byproduct of cellular metabolism. When cells make ATP, they generate carbon dioxide as a byproduct. So carbon dioxide is produced by the cells, it's released into the blood, and some of that carbon dioxide is exhaled in our breath every time we breathe. But as it turns out, carbon dioxide can react with water to form a weak acid called carbonic acid. Carbonic acid can dissociate into hydrogen ion and bicarbonate ion complex. And this is a very important reaction because this particular reaction determines our pH of our blood and body fluids. It's not the only one, but it's a very important one. So, as you know, being able to maintain pH within a range of normal values is important because we don't want to disturb cellular metabolism. We don't want to cause enzymes to denature. So, being able to get rid of excess carbon dioxide is very important in order to maintain a normal range of pH values. So we've heard a lot about oxygen and the fact that most oxygen that's transported around the body is transported through hemoglobin. A very small percentage is transported within the plasma, but most of oxygen is transported via hemoglobin. Well, as it turns out, carbon dioxide travels in 3 different ways around the body. About 70% of carbon dioxide is carried in the plasma, and it's carried in the form of a bicarbonate ion complex. So most of the carbon dioxide that's generated by cells travels in the plasma and travels as bicarbonate. A small percentage of carbon dioxide travels by itself, dissolved in the plasma. So roughly about 10% of carbon dioxide is found in the plasma. Another 20% roughly of carbon dioxide is bound to nitrogen in the form of amine groups. And basically, that means proteins. So a portion of CO2 is bound to proteins, of which hemoglobin is one type of protein. So generally speaking, the more carbon dioxide that is present in the blood, the higher the partial pressure of CO2. And that shouldn't be a huge surprise. But this relationship between carbon dioxide and water and carbonic acid forms what is called the carbonic acid bicarbonate buffer system. So it's one of several types of buffering systems that help us to maintain appropriate levels of pH in our blood and body fluids. And as you know, the levels of pH span roughly between 7.35 and 7.45. So let's take a look at this diagram here that also illustrates the role of carbon dioxide and its relationship within the carbonic acid bicarbonate buffer system. So let's look at the top figure here for just a moment. Carbon dioxide is generated from tissue cells. A little bit of that carbon dioxide can make its way into the plasma, and so it travels by itself within the plasma. However, most of the carbon dioxide can enter a red blood cell where it can combine with water to form this weak acid called carbonic acid. Some of those molecules are going to dissociate into hydrogen ion and bicarbonate ion. Now, some of the hydrogens are going to bind to hemoglobin. And that's a good thing because hemoglobin protein can act as a buffer. And if you recall the definition of a buffer is a molecule that can receive or discharge hydrogen ions. And so what it does is to help stabilize pH and prevent the pH from going too high or too low, because we're trying to keep it at roughly 7.4. That's kind of the ideal level for blood and body fluids. So hemoglobin can also act as a buffer to take up hydrogens. So bicarbonate ions can also swim around in the plasma as well. And these molecules also serve as a buffer, so they can take up hydrogens as needed in order to make sure that the pH does not go too low or become too acid. All right, so that's the carbon dioxide that leaves the tissue cells. So let's consider what happens when carbon dioxide rides around in the plasma and on red blood cell and makes its way back to the alveolus. So we have bicarbonate that has been dissolved in the plasma, Some of that can be taken back up by red blood cells, where it combines with hydrogen ions to form our weak acid carbonic acid. The carbonic acid can then be broken up into carbon dioxide and water, and the carbon dioxide can then diffuse out of the red blood cell, head back into the alveolus, where it can be then exhaled. So one way that the body helps to regulate pH is through respiratory functions. So in the next video, we'll talk about regulation of breathing and pH control.